Okay, so this is proteins, part two. When we first talked about this, we left off with this idea that the monomers of proteins are things called amino acids. So this is an example of one of those monomers where you see, again, central carbon. You see this functional group called an amine group on this side, and you see a functional group called the carboxyl group, sometimes called carboxylic acid group on this side, with this R group that is representing some other side chain, something else that really identifies which amino acid this is. So we're going to look at these just briefly again. Whoop, I got carried away. There you go. So here they are. Here's the different amino acids. There are 20 that we typically talk about here, with some of them being polar and aliphatic. Remember what that means. Non-polar and aliphatic, some of them being polar, some of them having positive charges. And when we talk about the charges, that generally is talking about what's going on the R groups. So we're looking at the typical R groups that are found on this amino acid. And then we have positive and negative ones, and we also have non-polar aromatic R groups. So we kind of walked through some of these before. You can see them over here in this diagram. I was trying to make this a little bigger if I could, so we could really see them a bit better. And there you go. So here it is. So look at the blue boxes to determine what the R groups are. So, and the way this is organized is great because it really tells you, basically it shows you what we're saying over here. So something that's non-polar and aliphatic, you can see as you look at these R groups here that many of them, because they're non-polar, have a lot of hydrocarbons in them. So you can see the CH2, CH3, CH3, etc. So that's a kind of a tip-off, as you know, if it's a hydrocarbon, has a lot of hydrocarbons, doesn't really have other functional groups on it that might associate or interact with water, then it's going to be nonpolar. And then knowing those definitions for what's aliphatic and aromatic is really helpful here. So we talked about this. We can also use symbols, and when we're looking at proteins, which are built by these long chains of amino acids, rather than writing each of those out entirely, there are some shorthand ways to do it. So we'll get a little practice with this. Just different shorthand or symbols that are used to represent a particular amino acid. This part here, though, is important that we know that there are certain amino acids that are essential. In other words, we have to get them in our diet. And I encourage you to look up what are some of the sources of isoleucine or leucine or cystatine to see what amino acid, where you find them, what foods you'd find them in. Here's the bonding piece. So we're going to have these amino acids. Here's one on the left and one on the right. As you can see, they've given you the simplest amino acid possible because these R groups are represented here just by H. And they're showing that when two of these come together, same thing as before, it's going to happen through dehydration synthesis. So look at where the water, here's the hydroxyl group on this carboxyl functional group is going to be taken out, and then this hydrogen that's on this amino group on the next door neighbor amino acid will be removed, and the result will be this, a peptide bond. So water would be part of the product too, so just don't forget that, but you can see how this linkage occurs. So this is a peptide bond, or very often we look at this as a peptide linkage. Now, why this is really good to just like burn this into your memory is that when you see this, it basically tells you right there the two amino acids are joined. So when you see a long chain of amino acids that build some protein, and I ask you, for example, how many amino acids are in that chain, what you would do is look for this, because this shows you right there that there are two right at this spot. So we'll see a few more coming up. A polypeptide is a big long chain of amino acids that are joined by these peptide linkages. And most proteins are large. They are polypeptides by and large. And you're going to see sometimes there's some things that are within those peptide bonds or within those amino acid polypeptides that are called non-peptide prosthetic groups. And I'll go into some specific examples of this. So proteins are extremely important to us and certainly in all of biology. So we'll look in some detail there. And this is that video you can look at in terms of what is protein synthesis, how are proteins made, and that comes later too. So this is where I think we really did leave off. We looked at the fact that once you form, that there are 4 levels possible of structure that can create a protein. So let's take a look at them one by one. So we have primary, secondary, tertiary, and quaternary. Primary structure is simply each of these amino acids that are in a chain that are held together by peptide bonds. So up here, you can see that by their abbreviations, each one of these circles is representing an individual amino acid that is bonded to the one next door by a peptide bond or peptide linkage. That's an example of primary structure. So that is the key to understanding primary structure, is knowing that it is the sequence of amino acids formed by these peptide bonds. Now, this example is showing that there's actually 2. There's one at the top that says it's the A chain, and the one at the bottom is the B chain. And the purpose of this is to show that in these amino acid chains or polypeptides can be formed like this by the cells, and then as they're forming something else, they can actually link together. So here you can see this linkage, and they're showing that this is an example of bovine or cow serum insulin. So cow would produce this type of structure for insulin. And if there's some little alteration that occurs anywhere along the way, like a mistake is made, the wrong amino acid is put in, it can totally change the ability of that protein to do its job. And we'll see this a little bit later. So the primary structure has to do with the amino acid sequence. Here's the example of a specific example of where something can go wrong when a normal hemoglobin, which is a protein, would have an amino acid at position number 7 on that chain, if you just counted, would be this amino acid, which is glutamic acid. But when someone inherits the gene that instead replaces that by another amino acid, this one being valine, that can cause sickle cell anemia. So it's the change in the beta chain of that hemoglobin. Now, if you're lost when I say beta chain, here's the example. Here's the alpha chain, this one up here, and this one would be the beta chain for bovine insulin. So we're talking about it in humans now. So there was a substitution that changed that hemoglobin molecule, which causes it to not be round, to cause the red blood cells, because that's where we would find these hemoglobin is within red blood cells. And notice that the red blood cells have some of them have this odd shape. So that's the sickle cell trait or condition. The secondary protein structure involves hydrogen bonds. Hydrogen bonds that will form once the primary structure has been made. So wherever hydrogen bonds can happen, you're going to see in some of these, I'll try to make this a little bigger. This is really ridiculously small. See if we can make it big enough that we can see it without blocking everything. So I'm going to move this over a bit. And if you look here, you can see, probably on yours you can enlarge it anyway. Here's an oxygen on this chain, this part of the chain below. there's a hydrogen. So you know already from water that this is a place where hydrogen bonding can take place between a highly electronegative atom like oxygen and hydrogen. So what that causes, though, is it causes one of two formations. One formation is like a pleat on your skirt. So some of these chains of amino acids can end up looking kind of like they've been pleated. And others, or even within the same long primary structure, some of them can form these spiral shapes. So you can see this over here. So this is called a beta pleated sheet. You can see that over here. And this is called an alpha helix. So beta pleated sheet, alpha helix, both of these are considered secondary protein structure. Here's just an additional picture of that. You can see that here. Let's go on. Now we go to tertiary. So we assume all the amino acids have bonded through peptide bonds, primary structure. Next, we have the hydrogen bonds forming, secondary structure. And now we're on to tertiary. So tertiary means that other things are connecting or joining or repelling. So all of the tertiary structure has to do with what are those R groups. Are they hydrophobic? Are they hydrophilic? Do they have a charge on them? What will they do? Do they have, in the case of certain R groups, do they have something, one of the amino acids called cysteine? And they can form an actual bridge across called a disulfide bridge, because if you look back at the amino acid cysteine, you're going to see that sulfur atom exists within its R group. So tertiary structure causes the protein to really take shape. You'll see lots of different possible interactions that can happen. You can have between part of these, so there'd be this would be the chain of amino acids, perhaps obviously with some other things we can't see here because it would get complicated, but primary structure is the chain, secondary structure, there would be some of this that would be pleated or spiraling, and then the R groups start to interact within this long chain, and that's what you see here. Pick out some of these interactions, look at them carefully, such as the ionic interaction here, hydrophobic interactions where notice this is kind of like hiding out. It's going away on the interior of this molecule away from water. Here's a disulfide bridge between two cysteine amino acids. Here's a hydrogen bond that's formed. So all of these things can further happen in tertiary structure. And then finally we get to quaternary. And in quaternary structure, we have several of these things that have all done all of the things we just talked about, and they form a larger unit of a protein, this quaternary structure. Not all proteins are quaternary in their structure. Some of them stop at the tertiary level, but you can see how this one would look. In this particular one, they're showing a prosthetic group. See this little thing in here that's called heme, which is representing iron. So in hemoglobin, we have iron, which is able to pick up oxygen and carry it around in our bloodstream. So here they are, the 4 different levels of protein structure: primary, secondary, tertiary, and for some, quaternary. So that's important to remember. And then we'll go a little bit further. We have just 1 minute. So if this protein structure is disrupted due to things like pH changes or change in temperature, then it causes the protein to change in its form. We call that denaturation. So this egg is a good example of a protein that has been denatured, and particularly this white, which is the egg white, is a protein. And when it's heated, notice what happened to it. It goes from a sort of an opaque or clear look to a solid white. And then we're on to nucleic acids. So that is, we'll stop with the egg, that is the finale on proteins.